Bismillahirrahmanirrahim, and welcome back to this next video. And in this particular video I want to focus on the test cross, which is an important technique in the field of the genetics. So have you ever wondered how the scientists or the geneticist they determine the genetics behind the visible traits like the flower color or the shape of the seed, that why some flower they are purple, why some of the flowers they are white, why the shape of the seed is round in some cases it is wrinkled. So how the scientists they determine the genotypes behind these visible phenotypes? So they use a technique which is known as the test cross, and that is going to be the focus of this particular video. So the first thing is, what is a test cross? So a test cross is a genetic cross between an organism showing a dominant phenotype and another organism which is homozygous recessive organism to determine the unknown genotype of the dominant phenotype. So what it means is that the test cross is going to tell you about the genotype. It is going to help you determine the genotype but not the phenotype. So you can determine the unknown genotype using the test cross. To understand the test cross, or to get the very basic concept of the test cross, you have to go back to like mid of the 1800s, where Gregor Mendel, who is known as the father of genetics, he was working with the pea plant and he was focusing on a lot of traits in the pea plant. For example, if you talk about the seed, he was interested in the forms of the seed, that whether the seeds they are round or they are wrinkled. He was interested in the flower color, that if the flower is white or if the flower is violet. He was interested in the color of the pod, for example, that you have got a yellow pod or you have got a green pod. And he was also interested in determining the height of the pea plant and the position of the flower as well. So he was doing you can say a lot of things, and he was using a lot of techniques. Now one of his key technique, that was the test cross. For example, Mendel saw a tall plant, but he didn't know if the tall plant has two alleles like capital T and capital T. Today we call this is the homozygous, that whether the tall plant is homozygous or the tall plant that contain one capital T, that contain one small t, two different alleles. Today we call this is the heterozygous. So he don't know whether the tall plant that he is getting, whether that is homozygous or that is the heterozygous. So he crossed it with a true breeding short plant. Now you need to understand the concept of the true breeding. When we use the term true breeding, that would mean that how many times you are going to say, for example, if you have got this small t short plant, so if you sow it once, all the plants they are going to be short in nature. If you sow them again you are going to get the short plant. So if you like do that like forever, you are going to get the short plants all of the time. This is what the true breeding short plant mean. Now this method of the test cross that allowed him to uncover the fundamental law of inheritance, including the law of the segregation. And the law of segregation, we have already seen that in the course, that what the law of segregation actually means. Now the test cross that like might seem simple today because we get the idea how the test cross works, but back then it was a revolutionary idea. And this revolutionary idea, it helped Mendel prove that the traits they are inherited as discrete units, what we now call is the genes. Let me give you some scenarios. For example, if you have got one parent that is, you can say, heterozygous style, I've got another parent again that is heterozygous style, and if you cross them, you are going to get this plant. This is style in nature, but this is homozygous style. At the same time, you are going to get this one is the tall plant, but this is heterozygous in nature. So how are you going to differentiate, or how are you going to determine that this that what is the genotype of this tall plant and what is the genotype of this tall plant? So you have to go through the test cross. Now what is the science behind it? The first thing you need to understand is the dominant versus the recessive trait. Dominant would mean that particular phenotype is going to express itself in the homozygous as well as in the heterozygous form, but the recessive phenotype is only going to express it in the homozygous form. It cannot do that in the you can say heterozygous form. For example, if you've got the tall plant, as I've told you, so with capital T and the small t, that would be a tall plant. Capital T and capital T, that would be a tall plant. So the tall plant phenotype is expressing itself both in the homozygous as well as in the heterozygous condition. But if you talk about the short one, it is only going to express in the homozygous recessive condition on the small t and the small t. That is going to give you a short plant, and that is true for all of the traits that you study when you talk about the Mendel's experiment or you talk about any of the genetic traits. And you need to understand the genotypes and the phenotypes. So genotype, that would be the you can say genetic makeup, and the phenotype, that would be the physical appearance, or that would be the physical interpretation of the genotypes. For example, this capital T and capital T, that is homozygous and that is dominant in nature, so it is going to give you a tall plant. This is in the homozygous form, it is going to give you a tall plant. Similarly, if you look at this one, the capital T and the small t, that is heterozygous, but is the capital T that is dominant over the small t, again it is going to express itself in the heterozygous condition, suppressing the appearance of the small t trait over here. So again you are going to get the tall one. If you talk about this one, this is homozygous recessive, and this is the only condition where you are going to get the short one. So here you can see there is a small t allele, but in the presence of the capital T allele it cannot express itself. Therefore we call this as the recessive, and we call the tall phenotype is the dominant one. Let us have some test scenarios. For example, you've got a parent, the genotype, that is capital T, that means it is homozygous. The phenotype is tall. It can only produce a single type of the gamete, and all of the gametes, they are going to carry the capital T allele. If you are going to cross them with the parent number 2, and again, as I've told you that in the test cross, if you are interested in determining the unknown genotypes, you have to cross that with a homozygous recessive parent. So this is the genotype over here, that is small t and the small t, that is, you can say, a homozygous recessive. Phenotypically it is short, and again it is only going to give you a single kind of the gametes, the small t. So all of the gametes that would carry the small t allele. So if you make a Punnett square for this, these outside of the Punnett square that are showing you the gametes of the parent, and the interior of the Punnett square, that is going to give you the fertilized product. So on one side, if you take the dominant plant, the tall plant, it is going to give you all of the gametes in the capital T and the capital T form. And the upper side, that is going to show you the gametes of the other parent, and again that is only going to give you a single type of the gamete, that would be the small t. So if you cross them, if you get the tall t from this parent, the small t from this parent, you are going to see a genotype like the capital T and the small t. Similarly over here, the capital T from here, the small t from here, again the capital T from here, the small t from here, and again the capital T from here and the small t from here. So what you are going to see is, you are going to see a genotype that would be heterozygous in nature, and the phenotype that would be tall, but 100% of them, they are going to be tall. So if in a test cross you are getting 100% of the dominant phenotype, that would mean that the unknown genotype, that would be homozygous, and that is going to be represented by this particular, you can say, Punnett square, because the small t, they are not going to express itself in the presence of the capital T. So this shows that if a parent genotype is homozygous dominant, that is going to express itself in like 100% of the offspring, and you are not going to see any of the recessive phenotypes. Let us look at the scenario number 2, that will you can say give you a very clear idea in comparison to the first one. Now the parent number 1, again that is a tall plant, but that is in the heterozygous condition. They have got the capital T allele as well as the small t allele. Now you are going to get two types of the gamete. 50% of the gamete, they are going to give you the capital T. 50% of the gametes, they are going to give you the small t. And again, as we are talking about the test cross, so the second parent, that would be homozygous or recessive, and short in the short phenotype. And in this particular case, again, you are only going to get a single type of the gamete. So this would be one difference. In the homozygous form, you are getting a single kind of the gametes in the dominant phenotype, but in case of the heterozygous one, you are going to see two types of the gametes. So if you cross them, again, this is the parent number 1. Now, the 50% of the gamete, they are showing the capital T allele. 50% of the gamete, they are going to show you the small t allele. Similarly, the other parent, of course, that is, you can say, the homozygous recessive form. So if you cross them, the capital T from here, the small t from here, the capital T from here, the small t from here. Similarly, the small t from here, the small t from here, and the small t is coming from this parent, the small t is coming from this parent. Now, if you look at the genotype, you are going to see two types of the genotypes. The heterozygous tall, the phenotype, that would be tall, but at this particular time, this is not 100%. This is only 50%, because 50% of the genotypes that you are going to see is the small t and the small t, and they are short in phenotype, and they are representing 50% of the offsprings. So if the unknown genotype, if that is heterozygous in nature, you are going to see both the dominant and the recessive phenotypes. And if the unknown genotype, that is homozygous dominant, that is going to show only a single kind of the phenotype, and that would be the dominant one. So using this particular test cross, you can determine that the dominant phenotype shown by a particular, you can say, trait, for example, in this particular case, tall or short, if that is homozygous dominant, that is going to express itself in 100% of the offspring, and all of the offspring, they will be showing the dominant phenotype. But if the dominant phenotype is in the heterozygous form, that is going to represent itself in 50% of the offspring, and the other 50 type of the offspring, that are going to show you the recessive condition. Now, why are we using the test cross? What are the applications of the test cross? Let me give you some of the examples. If in agriculture, if you are interested in the pure breeding or the true breeding plants, for example, you want all of the peas with the round seeds. So what you can do is you can determine the genotype of the plant. If they are true breeding, you can sow them again and again, and all of the time you are going to get the pea with the round seeds. If you talk about the animal breeding, if you are interested in an animal, for example, a cattle, for example, that is very massy, so you can use this test cross. In the research, if you talk about the human beings, for example, they are very important in studying the genetic disorders, like the autosomal dominant condition, the recessive condition. And if you talk in biotechnology and the genetically modified organisms, they are very important, because these genetically modified organisms, if you want to verify their stable inheritance, you can use the test cross. Now, there are some of the misconceptions in the mind of the student. They should be very clear at this particular stage. The test cross is not the same as the back cross. We will discuss about the back cross in the next video, but they are different from each other, though they are very much related. A dominant phenotype doesn't always mean a homozygous genotype, and we have seen that the dominant phenotype, it can express itself in the homozygous as well as in the heterozygous genotypes. And the test cross is not going to change the genes, it's only reveal them. Now this is quiz time. What you have to do is that if you have a yellow seed, which is a dominant phenotype, that is crossed with a green seed, which is a recessive phenotype, and all of the offspring that is yellow, so what's a likely genotype? So what you have to do is you have to make a Punnett Square, and I'm expecting your answers in the comment section. If I recap the thing, the test cross, that is a tool to determine the genotype. This is very important in both the classical and the modern genetics. It have got applications in the agriculture, in animal breeding, in studying the genetic diseases. And the phenotype, doesn't always tell you the full genetic story. We will continue our discussion in the next video.